Biology Tour of the Cell, directed by Team 23, Carlos, Karen, and Zach. In the video, we will be going over the animal cell, the plant cell, the bacterial cell, the nucleus, the endomembrane system, the cytoskeleton, and the mitochondria.
This is an animal cell. It has various organelles. Let's start with the ribosomes. The function of a ribosome is to make proteins for messenger RNA. It is made up of a large and small subunit that work together to make an amino acid chain. The mRNA is read and coded into a resulting amino acid chain. Proteins are this resulting nucleotide chain. Ribosomes are made within the nucleolus and the nucleus and can be found in various locations of the cell. In the cytoplasm, there are free ribosomes as opposed to bound ones in the endoplasmic reticulum. They are basically where the synthesis of proteins are crucial. Next, we move on to the Golgi apparatus and vesicle. The Golgi apparatus uses proteins from the rough endoplasmic reticulum, which then is transported from the Golgi apparatus. The proteins have carbohydrates added to them, which are enveloped by the cisternae. The cisternae are individual Golgi sacs. They help do the processes of distributing proteins with enzymes present. Not only does it package and send out proteins, but as well macromolecules. It does so by adding phosphates and carbohydrates to the macromolecules, which are then receivable by the tags added. The processes used are glycosylation and phosphorylation. The tags determine what goes where. In the Golgi vesicle, simply they are the sacs that stem from the Golgi, which package and process macromolecules, which tags and stores them for later. They are next to the Golgi apparatus. Next, we move on to the endoplasmic reticulum. The rough endoplasmic reticulum is given its name because it has ribosomes attached to its surface. It is attached to the cytosolic side of the endoplasmic reticulum. The ribosomes bound on the membrane are for translation in which proteins are created. It as well tags those proteins for a location in which for them to go. The proteins are packaged and sent to the Golgi apparatus for further processing. The rough endoplasmic reticulum is dispersed across the cell, but it is mostly in clumps around the nucleus and Golgi apparatus. The smooth endoplasmic reticulum is distributed fairly along the cytoplasm and is connected to the rough endoplasmic reticulum. This organelle has mostly to do with the production of lipids and the metabolism of them. Not only that, but through the circulatory system, can as well make steroid hormones for the endocrine glands in the adrenal cortex. Now we move on to the nucleus. The nucleus contains the genetic information of the cell. It has DNA that can be used for replication and reproduction. It has a double membrane layer called the nuclear envelope, which separates it from the rest of the cell. This layer is a phospholipid bilayer, just like the cell membrane, and it helps control what molecules leave and enter. The nuclear envelope is connected to the endoplasmic reticulum in order to transmit proteins into the nucleus faster. Like the cytoplasm, there is a nucleoplasm that is the jelly of the nucleus. Transcription happens in this cell, and the cell organelle is in the center. The nucleolus is within the nucleus of the cell. Its purpose is to make ribosomes. With the help of RNA, DNA, and proteins, ribosomes are facilitated in this organelle. What is made in this organelle specifically are the subunits that make up ribosomes. With the help of these subunits, proteins can be made through translation. Chromatin is condensed DNA in the nucleus alongside histone proteins. The chromatin's physiology concerning how wound it is determines which section of the DNA can be read. This is possible due to methylation and acetylation. If DNA is too condensed, it cannot be read. This is the result of methylation. Acetylation, on the other hand, is the opposite. And as for the mitochondria, we will talk about it later in the video. In animal cells, the cytoplasm vacuoles play roles in the endocytosis and exocytosis. In exocytosis, molecules like proteins and lipids are moved away from the cell, in which the vacuole itself carries it out of the cell in a bubble. The vacuoles act as a container for certain molecules and are a result of cell signaling. Vacuoles do the job of isolating certain molecules for different tasks, whether it is for degradation, excitation, maintaining waste, or other processes that require these organelles. They play an important role in the cell and are dispersed to the cell depending on the molecules that are needed for it to be present. The peroxisome secretory vesicles are similar to vacuoles in which they as well hold molecules, but instead break them down. These vesicles are small in the vacuoles and break down toxins within the cell. These are not like lysosomes that are formed by the Golgi apparatus and they themselves self-replicate. They are named after a product when they break down molecules. H2O2 peroxide is produced when they break down organic compounds, which is then broken down further into water. It is single membrane, so it does not have folds and uses oxygen to oxidize these organic molecules similar to the mitochondria. They are largely dispersed throughout the cell. I messed up. Lysosomes are small and contain enzymes that help the cell use nutrients and as well have a role in autolysis when needed. They help digest and have a phospholipid bilayer. The enzymes in the lysosomes are called acid hydrolases and work in low pH levels. They are important in which they hold potentially harmful enzymes away from the cell and maintain waste by removing unwanted molecules. The intermediate filaments are found throughout the cytoplasm. Its main function is structural and is to keep cell shape and accumulate them into tissues. The most important role of the intermediate filaments is to cause support in the plasma membrane when it comes in contact with other cells. It does not have to do with the movement of the cell, but instead has to do with the retaining structure. Microfilaments are proteins that are throughout the cell which have a role in the support of the shape and organization. They have a major role in cellular movements as well and are solely responsible for any movement. Microfilaments are responsible for the movement of the cell of any sort and are largely dispersed throughout the cytoplasm. The centrosome of animal cells have two centrioles that aid with the mitotic spindle in helping with cytokinesis. Plasmodesmata are holes in between plant cells that allow for the transportation of molecules. They are important in the cell as they allow for nutrients to pass through cells. This is important in distributing molecules like water, which allows for there to be an even distribution of where they are needed, as in the
plant
example.
cell.
The pores are present in the cell walls of the plants, which are holes of the phospholipid bilayer, and create the symplast pathway. They allow for cells to connect and send molecules to each other. Vacuoles play an important role in the storing of water. They are about 90% of the entire cell. They are enclosed by a membrane called the tonoplast. Inside the vacuole, there is a cell sap which consists of sugars, salts, and organic acids. The main function of the vacuole is to maintain turgor pressure, which allows for the cell to maintain its rigid shape. This is important as to when water either increases or decreases. I messed up. Lysosomes are small and contain enzymes that help the cell use nutrients and as well have a role in autolysis when needed. They help digest and have a phospholipid bilayer. The enzymes in the lysosomes are called acid hydrolases and work in low pH levels. They are important in which they hold potentially harmful enzymes away from the cell and maintain waste by removing unwanted molecules. The intermediate filaments are found throughout the cytoplasm. Its main function is structural and is to keep cell shape and accumulate them into tissues. The most important role of the intermediate filaments is to cause support in the plasma membrane when it comes in contact with other cells. It does not have to do with the movement of the cell, but instead has to do with the retaining structure. The chloroplast is one of the most vital organelles in the plant cell. This is the source of photosynthesis in the cell and is capable of creating sugar and oxygen. They, as well as the mitochondria, contain their own DNA and can reproduce autonomously, regardless of the nucleus. The DNA consists of has a code for proteins involved in its electron transfer chain, which can make redox reactions, and the proton motive force. It has two phospholipid bilayers with different processes in between each. Its layers consist of its outer membrane space, intermembrane space, and inner membrane space. Within the inner membrane space is a region called the stroma that consists of ribosomes, granules, thylakoids, and lumens, which all go in hand to make the processes of photosynthesis possible. These organelles are largely dispersed throughout the cell and are larger than the mitochondria. Similar to an animal cell, the plasma membrane of a plant cell controls what comes in and out of the cell. It is semi-permeable to molecules, meaning that it allows certain molecules and essential molecules to leave while waste exits. For example, carbon dioxide, water, and other small molecules are able to enter freely, while large molecules like amino acids are not. The membrane itself is to protect the structure of the cell and provide support. It adjusts in size according to water levels and surrounds the cell throughout. Although the plant cell already has a cell membrane, it as well contains a cell wall. Only plant and bacterial cells contain cell walls. Their purpose is not only for structure, but as well to transfer molecules. The plant cell wall forms an adoplast system, which allows water and certain molecules to pass between plant cell tissues. This is a result from the plasmodesmata organelles contributing to their apoplastic pathways and symplastic pathways. The cell wall has to do with distributing molecules across the plant cell. It surrounds the cell membrane and is usually made of cellulose. The Golgi apparatus is made of folds and vesicles that form in structure. It is in charge of taking care of proteins that are transported from the endoplasmic reticulum. The proteins that it attains are packaged and sent out of the organelle with a tag in the form of exocytosis. Not only does it do this to proteins, but to other macromolecules that come from the endoplasmic reticulum. As a result, this can be thought of as a post office of the cell that ships out molecules for the places that they are needed. The cytoplasm is a jelly. The endoplasmic reticulum is similar in the plant cell as it is in the animal cell. It has ribosomes attached to its surface, the roughy R, and is for translation in which proteins are created. The proteins are then packaged and sent to the Golgi apparatus for further processing. In the smoothie R, the organelle has more to do with the production of lipids and the metabolism of them. The filamentous cytoskeleton is for the structure of the cell, as well for organelles being able to interact with each other by moving along this skeleton and getting places where it needs to be. It is important in sorting organelles and helps with structural support. Ribosomes in the cytoplasm of the plant cell are largely responsible for the expression of genes within the cell by reading mRNA. The mRNA reading is done by the ribosomal complex with the help of tRNA, which aids the ribosomal complex in building a polypeptide chain. The tRNA's task is to attach itself to the corresponding code of genes present in the mRNA while it's in the corresponding binding site with the ribosome. Once finished, a protein is made. Basically, whatever the mRNA codes for, the ribosomal subunit reads and forms a corresponding amino acid chain for a macromolecule. The nucleus is one of the most important organelles in the cell, as it guides and tells the others what to do. This organelle holds the DNA of the cell. It regulates the synthesis of proteins within the nucleolus. It contains the nucleolus, and in this organelle is where cell division is made possible, as it itself must split. The DNA in the nucleus is important for the generation of new cells and for the making of proteins vital to carry out tasks, which as well makes enzymes. The nucleolus is inside the nucleus and has a process of translation from the template of DNA in the form of mRNA. With the nucleolus, proteins can be made, and as a result, rRNA, tRNA, and ribosomal subunits can be made. This is essential to the making of certain genes required for the cell. The ribosomal subunits that are made in the nucleolus are required to translate mRNA with the help of tRNA, making the amino acid chain. The nuclear envelope is responsible for keeping the nucleus and nucleolus away from the rest of the cell. It has a porous membrane that allows material to both enter and leave the nucleus. Plant cells have both a chloroplast and a mitochondria. The chloroplasts are needed for photosynthesis. The products, sugar, water, and oxygen, are reactants that are needed for the mitochondria, which in turn creates ATP. So basically, the products of photosynthesis are used as reactants for the chloroplasts. The structures in the plant and animal cells are all similar, but they differ only in 3 organelles. The plant cell has chloroplasts, cell walls, and vacuole organelles. Vacuoles are present in animal cells as well, but in the plant cell, they take up a large amount of space, while in the animal cell, they do not.
The bacterial cell. In the bacterial cell, we have several organelles involved. We have the plasma membrane, the nucleoid, the capsule, the cell wall, the mesosome, the ribosomes, and the cytoplasm. Above which, for movement, we have the bacterial flagellum. Let's start with the cell wall. Now, the cell wall provides our bacteria with benefits ranging from protection to prevention from bursting under osmotic pressure. Let's move to the capsule. Now, the capsule is considered a virulence factor in our bacteria because it enhances its ability to cause diseases. Above which, the capsule protects it from engulfment from other eukaryotic cells, and capsules also contain water, which protects the bacteria from desiccation. Take a look at the nucleoid over here. As you may notice, the genetic material of this bacteria does lack a nuclear membrane. Now, this nucleoid portion is also known to contain RNA and proteins and enzymes that may be used for other cellular processes. Now, let's come to the plasma membrane over here. The plasma membrane provides protection to the cell and allows for movement across the membrane. This membrane is a phospholipid bilayer. After which, let's take a look at the mesosomes. Now, the mesosomes are structures found in the prokaryotic cell that allows for cellular respiration to take place. Let's move on to the ribosomes over here. Now, the ribosomes are present free in the cytoplasm, and these aid with the translation of mRNA to proteins. Now, take a look at the cytoplasm over here. The cytoplasm is this gel-like matrix where the functions of the cell take place, such as cell growth, metabolism, and replication. Now, as you may notice, there's this tail-like structure at the end of our bacteria. Now, this long, thin structure is used for movement. It ranges from 0.1 micrometers to 50 micrometers in diameter, but usually average around 2 micrometers. Now, after this, let's take a look at the nucleus in heavy detail. Now, you may notice that there are several thread or string-like structures in our nucleus over here. These guys are known as chromatin. Now, we have 2 types of chromatin. We have euchromatin and heterochromatin. Euchromatin is an active form of chromatin that is acylated and it is allowed to be transcribed and used in the central dogma to help make proteins. At the same time, heterochromatin is an inactive form of chromatin that is methylated, that is not involved in making proteins. Now, let me elaborate on euchromatin. Now, I mentioned a few seconds ago that it was acetylated, as in the histones of this part of the DNA was combined with an acetyl group and allowed to be expressed, as in it was loosely tied. At the same time, heterochromatin, when it was methylated, it was tightly packed, and as seen, it's in tiny, compact rings of DNA. Now let's move to the nuclear envelope. Now this nuclear envelope is unique because it is only found in eukaryotes. Now this nuclear envelope over here surrounds the nucleus with a double membrane with several pores. Now these pores regulate the passage of several macromolecules such as proteins, mRNA, tRNA, but permit free passage to water and ATP. Without such pores, it would be really difficult to conduct simple cellular processes such as the central dogma. Better yet, we won't be able to replicate our DNA. Hence, these pores are absolutely vital to the survival of this cell. As you may notice, between our two nuclear envelope membranes, we have this dense, fibrous network known as nuclear lamina. Now, the nuclear lamina provides mechanical support and is usually very important in the cases of DNA replication and cell division. Now, as you may notice over here, we have this big creamish colored spot dead in the center of our nucleus. This is known as the nucleolus. It's a round body located inside the nucleus, as proven by the diagram of a eukaryotic cell. Now, this nucleolus helps make the subunits of the ribosomes, the large and the subunits namely, and these subunits help form proteins. But in order to make any type of proteins, we're going to need to transcribe the DNA. But our DNA is long and has several parts that are not needed. Those are known as the introns. Now, these introns are cut out by chemicals and substances in our nucleus known as spliceosomes. These spliceosomes are large, complex molecular machines found in the nucleus of eukaryotic cells.
In the endomembrane system, there is the nucleus, the rough-ER, the nuclear envelope, nuclear pores, ribosomes, smooth endoplasmic reticulum, secretory vesicles, lysosomes, Golgi apparatus, and the plasma membrane. As stated before, the nucleus has to do with the transcription of genes and contains DNA. It as well directs cell activity through the expression of genes. tRNA, mRNA, and rRNA are formed from nucleic processes. The nuclear pores help facilitate movement of molecules in and out of the nucleus, such as proteins. The nuclear envelope separates the nucleus from the other organelles. The endoplasmic reticulum, smooth and rough, help package proteins and produce lipids. Ribosomes facilitate protein synthesis. Secretory vesicles and lysosomes are involved in waste management and digestion within the cell. The Golgi packages and sends information from the endoplasmic reticulum to the other parts of the cell, and the plasma membrane regulates molecules entering and leaving the cell.
A protein that is destined for exocytosis is made like any other protein, where DNA goes through the central dogma, which is eventually translated into a protein. Now, this protein is packaged by the Golgi apparatus in consideration with the extracellular fluid that contains the cell around it. At the same time, a transmembrane protein is a protein that's made in consideration with the cellular membrane, as in that it has hydrophobic and hydrophilic regions accordingly so that it can apply itself to the membrane and function appropriately. As mentioned earlier, the lysosomes are made by the Golgi body. When the Golgi body is met by a transport vesicle sent from the rough ER, the body itself is triggered and a series of reactions cause release of a bubble of proteins that is, well, known as the lysosome.
The cytoskeleton has three components, and those are the microtubules, microfilaments, and intermediate filaments. A microfilament is a double helix of actin monomers. Microfilaments are the things responsible for the biconcave shape in red blood cells. Its major functions are cell shape and support, cell movement, cell division, which is cytokinesis, vesicle transport, and muscle contraction. Microtubules are hollow tubes made of tubulin dimers. One alpha tubulin and one beta tubulin combine to form a tubulin dimer, and those tubulin dimers are assembled to form the microtubule. Many organelles are tethered to microtubules, which means that microtubules guide the arrangement of organelles in the cell. Its major functions include cell shape and support, cell movement by cilia and flagella, cell division, vesicle transport, and lastly, organelle movement. An intermediate filament is a polymer of proteins which vary according to cell type that combine to form strong cable-like filaments that provide animal cells with mechanical strength. Intermediate filaments bind to cell junctions called desmosomes, forming a strong interconnected network. When there is a defect in the intermediate filaments, the resulting disease is usually epidermolysis bullosa, which is the disease that causes the skin to be very fragile, causing the skin to blister and tear easily. Microtubules and microfilaments both become longer by the addition of subunits, and both get shorter by the loss of subunits. At high subunit concentration, the subunits can be added more quickly, but one side of the microfilament or microtubule builds quicker than the other. That side is the plus end, and it assembles quickly, while the minus end assembles slowly. Motor protein kinesin, which carries cargo, such as vesicles towards the plus end, and motor protein dynein, which carries cargo towards the minus end, interact with microtubules and microfilaments to cause movement. Actin is a protein subunit that makes up microfilaments, used by both striated and smooth muscles to contract and generate force.
Let's take a look at this example of endocytosis. Now in the image we have a pea-shaped cargo or any type of substance hitting the cell membrane, the phospholipid bilayer. Now usually we initially assumed that it would just bump into it and like a bubble form around itself and fall into the cell where the cell may do what it may please with the cargo. But over here we have details declaring that it is not as such, and we have several other factors involved. So when we have the cargo hit our cell membrane, it attracts clathrin and adapters that help pull the cargo in. And when the cargo is reached far enough, we have scaffolds and other adapters that help cut off the end of it. Now, to effectively cut off the end of the cellular membrane and form a full bubble of what we have picked up, we have a scissors effector. Now, the scissors effector uses actin filaments as if it were glue or it were a stapler and connect our bubble together, allowing the cell to do as it may please with whatever it has picked up. Now, we have two different types of endocytosis. We have phagocytosis and pinocytosis. Now, phagocytosis is when the cargo is one big chunk. At the same time, pinocytosis would be several little chunks that come together and push against our cellular membrane to yield a very similar result. Now, this bubble, when it comes into the cell, is known as the vesicle. Let's finally move on to the mitochondria.
The mitochondrion has many parts to it that makes this organelle be able to produce as much energy as possible. Without this organelle, we wouldn't exist because we wouldn't have near as much energy that we need, which cells use to stay alive. Mitochondria has its own DNA. The genes in the mitochondria's DNA is used for making the complexes that are included in the electron transport chain. Ribosomes are involved in translation of proteins or complexes for the making of energy. DNA is different in mitochondria because the DNA is specialized for its function, which the DNA makes complexes that are used for making ATP. A mitochondria has two membranes, which is important because it needs to maintain a concentration gradient, which this concentration gradient is a proton motive force that creates ATP by the pumping of protons through the ATP synthase pump. The outer membrane has porins that allows small molecules such as some sugars that allows for small molecule concentration to be similar to that of the small molecule concentration of the outside of the mitochondrion. The inner membrane is folded to increase surface area to maximize how much energy can be produced, since more complexes can be attached due to more surface area. This allows for the buildup of protons to build the proton motive force that drives the synthesis of ATP. The cristae in mitochondria is a fold in the inner membrane that increases the surface area, which is important for maintaining a proton motive force. The ATP synthase pump is a pump and rod that rotates to form ATP when a proton is pumped through it. This adds an inorganic phosphate to ADP. The matrix in the mitochondria has a phospholipid bilayer that makes the concentration different from the outside of the inner membrane, which is the sole reason why ATP synthesis can occur. Granules in mitochondria connect inner and outer membranes to maintain homeostasis. Endosymbiosis is a theory that suggests that archaebacteria ingested a bacteria via phagocytosis that was very similar to a mitochondria that, instead of digesting the mitochondria, utilized it to make as much energy as possible. Evidence for this is the fact that mitochondria and chloroplasts have their own DNA and ribosomes that make their own needed proteins for their function. Also, mitochondria and chloroplasts showed up for the first time at the same time as eukaryotes did. Although this could be a coincidence, it is highly unlikely that these structures showed up at around the same time.
To recap, we went over the animal cell, the plant cell, the bacterial cell, the nucleus, the endomembrane system, the cytoskeleton, and the mitochondria.